In the last video clip, we talked about how specific immune responses can be divided up into 4 categories, depending on whether the immunity is gained through a natural process or an artificial process, and then whether the immunity itself is an active form of immunity or a passive form of immunity. In this video clip, we're going to be talking about passive and active immunization. So an immunization will be a medical procedure. So we're going to be talking about artificially acquired active immunity and artificially acquired passive immunity. And again, the difference between active and passive, when you have active immunity, it means your own immune system has generated a response and you have memory to an antigen that you've been exposed to before. Passive immunity is where you get large doses of previously made antibodies when you don't have time to wait around for your own immune system to respond because your life or health are in serious jeopardy. All right, so let's talk about these artificially acquired passive immunizations. Now, first of all, these occur much less frequently than artificially acquired active immunizations. Artificially acquired active immunizations are what we call vaccines. And so we've all had those, I hope. Passive immunizations are really given generally in emergency type situations most of the time. And again, this is where a patient is given large doses of pre-made antibodies. And these pre-made antibodies sometimes come from human donors, animal donors. There are newer methods using biotechnology processes where they're being made in a laboratory type setting. So not as common. These are generally given intravenously or IV. And these are often antitoxins or antivenoms. Antivenin is another term for antivenom. So, again, if you were in a situation where you were bitten by a rattlesnake, you might be given, here in Alabama, you might be given the southeastern United States, you might be given this crofab, which is a combination antivenom for eastern rattlesnakes and copperheads and cottonmouths. So the 3 main type of poisonous vipers that might bite you in Alabama. Won't work for a coral snake. We don't have coral snakes up in this part of Alabama, but down in South Alabama they do. And coral snakes, you need a separate antivenom that works against those. So if you were unfortunate enough to be bitten by one of those snakes, you don't have time to wait around for your own immune system to make antibodies that will attach to that venom and neutralize the venom and help clear it out of your body. So instead you're going to have to get large doses of pre-made antibodies. There are some pitfalls with this, and from what I understand, they don't always give this right away because a number of patients will have an allergic reaction to it. And so they kind of wait around, I think, for a little while and see how you're reacting to the snake bite before they give that to you. Another example, rabies shots. So I know when I was a kid, like the worst thing you could possibly imagine having were rabies shots. If you got bitten by an animal and didn't know for sure whether it might be rabid, you might have needed to receive rabies shots. And what rabies shots were, they're not like the vaccination your dog or cat gets to help prevent rabies. That's a typical vaccine or immunization. This is where they give you human rabies immunoglobulin. So pre-made antibodies that will, in large doses, that will attach to the rabies virus and help clear it out from your body. And that's because rabies is almost 100% fatal. If you become infected with that virus, your immune system is not going to be able to clear it out in time to prevent that virus from killing you. So you have to get these pre-made antibodies. And this comes from human donors. So some immunoglobulins like that come from other sources. And when I was a kid, the rabies immunoglobulin was given IP into the intraperitoneally, so into the peritoneal cavity. So there were, when you're kids and you hear these rumors and so forth, these long needles that are going to go into your stomach, and it's extremely painful. I don't think they do that anymore, but at that time, that was how you had to have the rabies immunoglobulin delivered to you. All right. Now, how about active immunization? So this is what we call vaccines. This is what you're more familiar with. We've all had these, again, I hope. And vaccines artificially introduce you to the antigens of a pathogen without you actually being exposed to the pathogen, and this stimulates your own immune response and memory. So when you really do get exposed to it, you're going to have a secondary type response. You're going to rapidly respond, generate antibodies, generate a population of T cells that will help you fight the infection as well. Vaccines have to be very thoroughly tested, so there's a very high level of safety required. So don't believe a lot of the hocus pocus you hear about how unsafe vaccines are. Vaccines probably have one of the highest bars that they have to cross before they can be given to people, because you are giving people vaccinations as a preventative. They're not actually sick. You're trying to prevent infection. And so you can't give something like that to patients unless the pros far, far, far outweigh the cons. An ideal vaccine would not require numerous boosters. So you guys know how some vaccinations require that you go back more than once and get a repeat follow-up. Those are called booster shots. Ideally, a vaccine doesn't require that, but it just depends on the nature of the vaccination. We're getting ready to talk about some of the different types of vaccines that are available. But some vaccines, the primary response is so good and the memory that develops from it is so good and so strong that you don't need any boosters. But others, the immunity that's generated, maybe it's only limited to antibodies and not really T cells, or maybe the memory that's generated isn't all that long lasting. So those types of vaccines, those are the ones where you have to go back and get boosters to try to increase the memory that your immune system has to that particular antigen. And we'll see vaccines can be classified as either whole cell vaccines, or in the case of viruses, whole virus vaccines. So you're getting the whole thing, or subunit vaccines. A subunit means you're just getting antigen, an antigen or antigens of the pathogen. You're not actually getting a full cell or a full virus delivered to you artificially. All right, so let's talk about these whole cell or virus vaccines. And they come in a couple of different categories. You guys can read through more of the details in your textbook. There's some nice tables in Chapter 13 that summarize the differences, and you do want to be familiar with this information. Okay, first of all, some vaccines are either killed cell or an activated virus. So, for instance, there are some vaccines for bacterial pathogens where you're getting bacterial cells, but they're killed. Now, their 3D structures have been preserved so that your immune system will detect and react to the antigens, but they're not a living cell. Same thing with viruses. You can receive a whole virus, but it has been inactivated, more or less preserved, frozen in place with its structure so that it maintains all the 3D shapes of the antigens so that your immune system will respond to it properly. And how do they do this? They use heat, chemicals to kill or inactivate these microorganisms. But again, you're trying to preserve the 3D shapes of the antigens so your immune system will see those as authentic antigens. One thing they use, now maybe you guys have heard before that there's formaldehyde in vaccinations. It's one of the things the anti-vaccination crowd likes to use as a pitfall of having your children vaccinated. And yes, formaldehyde can be used to inactivate some viruses to prepare those vaccines. But if you guys remember when we talked about formaldehyde back in Chapter 9, we mentioned about how it's a cross-linker. So it will cross-link, cause adjacent organic molecules to bond to each other. And because of that, it's a great preservative. It locks three-dimensional structures into place. But once you treat something with formaldehyde and you lock all those three-dimensional molecules in place so you're preserving that 3D structure, you don't need the formaldehyde anymore at that point. So they purify the formaldehyde out. So they're not injecting your children with formaldehyde when that happens. So there's lots of bogus information out there from the anti-vaccination crowds, and you really have to do your own research from good, reputable sources and make sure you understand. Don't develop these dogmatic opinions about things unless you understand some of the scientific information behind it, some of the arguments that are being used. Same thing you'll hear about the poliovirus vaccine being grown in monkey cells, and they're injecting monkey cells into children. That's not true. Yes, the virus can grow in monkey cells in cell culture, and the virus can use those monkey cells as a host, but they're not injecting the monkey cells into children. They're purifying the virus out and injecting that instead. So you can't believe all of these kind of wacky things that you hear from the anti-vaccination crowd. All right. Now, some vaccines are live attenuated cells or viruses. In this case, you're actually getting a live bacterial organism or a replication competent virus, but they have been attenuated. So attenuated, that's an important term to learn when we're thinking about vaccinations. What that means is these attenuated bacteria or viruses, they still have antigens that your immune system can recognize, but they're no longer capable of causing disease. So they've lost their virulence factors, and they're no longer going to be pathogenic in a healthy individual. However, when you get this type of vaccine, those inactivated viruses or bacteria, not inactivated, those attenuated viruses or bacteria are actually going to establish inside you for a little while and replicate and stimulate this immune response. But then eventually your immune system is going to wipe them out and they're going to go away. These are generally great vaccines because they're going to trigger good B cell responses and T cell responses. Some other types of vaccines might only trigger a B cell response and not very good T cell responses. You get good memory from these types of vaccinations usually, and so they're generally really good. Now there are some cons. Sometimes people who are immunocompromised, they have weak immune systems, they may actually get sick in some cases from attenuated vaccines, so you have to be very careful with that. And also, unfortunately, we don't have attenuated strains of every virus and every bacterial pathogen out there. So, we only have these available for some things. And you can imagine the safety testing. If you're taking something like the measles virus and developing an attenuated version that doesn't cause the measles, you have really got to do extensive safety testing on that and show that it's not going to make the vast majority of people sick. With vaccines, you've always got to make sure the costs far outweigh the benefits far outweigh the cost. The pros have to outweigh the cons in order to use them. All right, how about subunit vaccines? So this is where you're not getting the whole bacterial cell or the whole virus. You're just getting an antigen or antigens, molecules of the pathogen, like maybe a particular protein or a glycoprotein, or maybe 2 or 3 different proteins or glycoproteins that you would find on that bacterial cell or virus or some other type of microorganism. So you do develop memory to this, because when you get exposed to the actual pathogen itself, you'll be able to generate antibodies and so forth to the molecules that are on the surface of the organism. A couple of good examples: the pneumococcus vaccine that's given for Streptococcus pneumoniae, which is a pathogen that can cause pneumonia and also causes meningitis in children. Is that type of vaccine. The hepatitis B vaccination that almost all healthcare workers have to get is an example of that too. So you're just getting antigens of hepatitis B virus. There's no attenuated hepatitis B virus strain that can be used for vaccination. We just don't have a safe one available. So instead they give you this subunit type vaccine. The pertussis, there are newer versions of the pertussis vaccine. Bordetella pertussis causes whooping cough. And there's an older version of the vaccine that was a attenuated bacterial cell, and or maybe it was whole, maybe it was an inactivated cell. I'd have to go back and look that up. But now there's a newer version that is a subunit vaccine version. All right, let's see. Hang on. Here we go. Moving on. So there's a new type of vaccine that's under development still, and these are called DNA vaccines. So these may wind up being given more in the future. So these are kind of interesting. So what they do, this uses biotechnology type processes, and they take plasmids, little circular pieces of DNA that naturally exist in certain types of bacteria, and you can grow up bacteria in the lab that will replicate those to very high levels. And they use gene cloning techniques. So they'll take the DNA gene that codes for an antigen of a pathogen, DNA of a pathogen, and they'll stick that artificially using gene cloning techniques into plasmids. And it's pretty easy to grow large amounts of plasmid DNA in a laboratory inside bacteria in bacterial cultures, and then you purify those plasmids. And then interestingly, when you inject DNA into skeletal muscle, some of the skeletal muscle cells will uptake the plasmids, which is pretty interesting because DNA molecules are big, so this is kind of odd that they'll artificially take up the DNA like that. And it's not a very efficient process, but you don't need a whole lot of your own muscle cells to do that. And once your cells do that, you got the DNA, the gene, the blueprint coding for the antigen. And so your cells start making the antigen, and they send it up to the surface. They display it on the surface, and that will stimulate an immune response. And so this is a newer type of vaccination, and down the road you may wind up giving your patients DNA vaccines. All right, how about toxoid vaccines? So this is another category that's pretty interesting. And back in Chapter 11, we talked about bacterial exotoxins and how very often they're responsible for the damage associated with the bacterial pathogen. It's not really the bacterial cells themselves. And so in this case, it's beneficial to have a vaccine that stimulates immunity against the toxin. So you make antibodies that can attach to that toxin and neutralize it and prevent it from causing damage. So the vaccines that do this are called toxoids. And so what they do is they take the authentic bacterial exotoxin and they modify it. They change it a little bit so that it still has the same epitopes, the same 3D shapes that stimulate your immune system, but it's no longer capable of causing the damage that the toxin does. So it's a modified toxin that can still stimulate an immune response. And some good examples of this are the vaccines for tetanus and diphtheria. Because both of those, remember, tetanus, the problem with that is the tetanospasmin exotoxin that we learned about in Chapter 11 that causes lockjaw. And then diphtheria, there's a toxin that causes a pseudomembrane to form in the back of the pharynx that can cut off the airway. So the problems with those two diseases are associated with that exotoxin. So the vaccines are designed to stimulate the production of antibodies that can attach to those toxins and neutralize them before they're capable of causing such horrible damage. You actually get vaccinated when you get your DTaP shot. That's diphtheria and tetanus and pertussis. And so you're receiving these toxoid vaccinations in combination for both tetanus and diphtheria, along with your vaccination against pertussis, which causes whooping cough. This is a box of tetanus toxoid. And notice here on the box, I wanted to highlight this word adjuvanted, because you may see that if you're working with patients who are getting vaccines. And this is something else that the anti-vaccination crowd gets upset about. But adjuvants are things that have been added to the vaccine formulation that have helped preserve and it's kind of a catch-all term for things that might help preserve the antigens, and or when they inject them into you, there are things included that help provoke stronger inflammation and a stronger immune response. That's why your arm can get really sore after a vaccination, because they've actually included stuff in there to try to provoke stronger inflammation and try to boost that immune response, even though it doesn't feel too good when it happens. But anyway, there are some anti-vaccination people who don't like the inclusion of the adjuvants. They think those are artificial chemicals, and they're doing all kinds of horrible things to children and so forth. But all these things have been tested very extensively. So, like I said, don't believe everything you hear from unreliable sources. All right, here's a question for you. Now, we've all heard of tetanus shots before, and we just talked about over here the tetanus toxoid. So when you hear about somebody having to get tetanus shots, is that artificially acquired active or passive immunity? And so now we've talked before about, here's an image of Clostridium tetani bacteria. They kind of look like spoons. The little orange areas are endospores. Remember, Clostridium is a genus of endospore-forming bacteria. So Clostridium tetani is everywhere out in the environment. It's an anaerobe, and it's one of those things. It's the rusty nail bacterium. You step on a rusty nail. The rusty nail may be covered in Clostridium tetani, and that puncture wound creates a little anaerobic area where the Clostridium tetani bacteria can grow. And the problem is not the bacteria, it's that the bacteria secrete tetanospasmin, an exotoxin, which inhibits skeletal muscle relaxation. It causes your muscles to lock up into a contracted state. This is a painting from a long time ago showing a patient with an extreme case of tetanus. So this doesn't happen too much more these days. All right, sorry, my phone's ringing off the hook. This is what you get when I'm doing raw, unedited video. But anyway, back to this. So you probably wouldn't see a case of tetanus like this in the United States these days, because if somebody begins to develop these types of symptoms, they're going to be treated. All right, so when you get your DTaP shot as a kid, that includes this tetanus toxoid. That's artificially acquired active immunity. You don't have tetanus, you haven't been exposed to it. You are receiving a vaccination so that if and when you do get exposed to Clostridium tetani, you are capable of very rapidly, because of your memory cells, cranking out tons of antibodies that will mop up that tetanospasmin toxin and get rid of it. But let's say you do step on a rusty nail and you don't get it checked out, and you start experiencing symptoms of lockjaw. So this is a different story. So in that case, you know this tetanospasmin toxin is already circulating throughout your body and is probably increasing in concentration, and that's an emergency situation. So in that case, you have to get a passive immunization with anti-tetanus antibodies. So you're going to have to be loaded up with not the toxoid, you're going to be loaded up with the antitoxin, the antibodies that will go around and attach to this tetanospasmin and help clear it out from your body. So there are technically two different types of tetanus shots. Usually when people say, I got to go get a tetanus shot, they're talking about the toxoid vaccination, a preventative. But in a case where you might already have some symptoms of lockjaw going on, then you're likely going to have to get those pre-made antibodies of passive immunization. Now, sometimes, for instance, one time back when I was in grad school, I was bitten by a lab rat, and that's another way that you can wind up with tetanus, because they have dirty teeth that might have Clostridium tetani on the surface. And so I had to get a tetanus shot in that case. However, I was still getting the toxoid at that point. They were just trying to give me a booster to try to go ahead and stimulate me to make my own antibodies just in case I was going to wind up with a tetanus infection from that lab rat bite. Because it does take a while. You know, it takes 3 to 21 days, averages 8 days after exposure to begin to develop those lockjaw symptoms. So, if you just need a booster to make sure you are making your own antibodies as a precaution, they can go ahead and give you that toxoid. All right, so that's kind of an overview of passive and especially active immunizations. The last video lecture here for Chapter 13, I lied. I think I said earlier that one was the final one for Chapter 13. We got one more. We're going to talk about the importance of vaccination and some critical concepts that everybody needs to understand about them.